membranes and G protein coupled receptors, drug targets.
Okay.
50% drug targets are GPCRs. They work on GPCRs and how they interact with the cell membrane. And our work is quite novel. We are the first in the world to show that the membrane interacts with these targets.
Good.
So it was both basically.
Sir, I work on stress proteins, heat shock proteins.
You told me.
Yeah, there's very varied number of proteins and all. While I was working earlier days with the biophysical aspects and all, we looked at the structure of the proteins, and we find that some structural change improves their activity significantly. So we did a lot of molecular biology studies and other studies. Now we made engineered proteins which are more active than the normal molecules, which are about one molecule is about 4-fold active, another is 7-fold active molecule. We patented these molecules. We want to use them for therapeutic applications. But to deliver a protein is a very big challenge because it will be digested or it will cause immune reaction. We are now using nanotechnology approaches to try and see how we can deliver that. So that is going to be useful in cardiomyopathy as well as Alzheimer's disease and eye diseases. Then we are working on cancer models, and we have one drug molecule which is working very effectively against retinoblastoma. We are doing genomics and proteomics. Then we developed a photodynamic molecule which can be used for eye treatment as well as cancer treatment. The mission of the institution is to conduct basic research. This is one thing which I was emphasizing. All CSR labs are set up with a particular technology in mind, chemical technology, aviation technology, things like that. But this is set up for a basic research in front and multidisciplinary areas of work, modern biology, plus of course seek potential applications of the work. This is a charter. I'll skip that. Yeah, and this is when the institution was inaugurated. At that time, we had many Nobel laureates, very senior people came. One of the person called Mike Crompton. He was there at the time, and he.
Our institutions are very similar. We both are committed to pursue basic research. We both have a firm belief that without basic research, it's not going to be possible to solve the current profound problems of human disease and human need. When I left London some days ago to come to Hyderabad, I explained to my colleagues that I was going to the dedication of the jewel in the crown of Indian biological research. It's interesting, isn't it, that Dr. Becker and I, quite independently, have chosen to see this centre as a jewel in an Indian crown.
So we are very proud of that, and we actually become a jewel in the crown. So you can see with other things. Our strength is we have.
पौधा लगाया ना अपने बारे में लगा हुआ है ना
Yeah, उनका भी पौधा है. Yes. Then we have very strong, good, outstanding pool of scientists. We have infrastructure facilities comparable to the best that I was just mentioning. So there is no problem about that. We have well thought of founding principles. One is excellence and the centralized facilities and resource sharing. These are three major points that we have strength. And then we have CCM is recognized as the Center of Excellence by UNESCO. Yeah. It's not only national level; it's actually global net as an international level. UNESCO recognized it as a Center of Excellence. And then CSIR Technology Award for developing DNA fingerprinting. Then another CSIR Technology Award for developing a process for polypeptide proteins to be produced. Another CSIR Technology Award for assay system for cancer drugs. Another CSIR Technology Award for universal probe. This is a very interesting concept.
जिस चीज़ के लिए नहीं मिला वो दिखा दीजिए
नहीं मिला तो दिखा सकते हैं. No, this is a very interesting thing. If an animal meat is there, from a piece of meat we can tell what animal it belongs to. So DNA fingerprinting, that is the one which is being used for crime detection and things like that. And then IFICI also given outstanding award. And World Academy of Science, the third World Academy of Science designated CSIR as a South Center of Excellence.
Nobel Prize.
Huh?
Nobel Prize.
Nobel Prize. Then this year we got the CSR Award for Innovation in Rural Technology. Dr. Ramesh Sunti just now described he has a new variety of rice. That rice now we popularized into the villages, and rural people are getting actually economic benefit out of that. So, but in recognition of that, translation of the work actually to the rural field and helping them, that's what we got this recognition for. And then, as I mentioned, we have no departments, around the clock functioning and pooling of resources, free access to all infrastructure facilities. And these are the unique features. We also have platform interaction now of arts where you're coming, also we did not show you. When we go out, we'll see nice art pieces are then including MF Hussain, other people artists. We had a camp. At the time, artists came here and they painted. Those paintings, some of them are with us. And then we have major facilities here. Some of them we will go through, see, and I'll just skip this because to just save time. And as I mentioned to you, these are the earlier former directors, and they are all recognized. One is Padma Bhushan, Dr. Bhargav is Padma Bhushan, is the founder director. The next director was Professor Balasubramanian, who has also received Padma Shri, and after that Dr. Laji Singh also received Padma Shri. Then all these people, all of us received Shanti Swarup Bhatnagar Award prize. You know, many laboratories don't have this many Shanti Swarup Award winners.
These are all people from this institute?
Yes. Now I'm here.
Some of them in the room.
Ghanshyam Swarup is here, Ramesh Sotti is here, Amit Chattopadhyay is here, Nagaraj is here, Shankar Narayana is here. Shankar, you just met the CDFD director, you met him, and these three are in different places now. Okay. Bottom four people are in different places, but they have got the award when they were working with us at CCA.
Price of success.
Yeah. And then one Infosys award, Dr. Imran Siddiqui got Infosys award. We have 8 J.C. Bose fellows and other fellows. And we have good papers in Nature, Cell, and things from the beginning of this year.
How many publications in last 28 years?
The last 28 years will be about 100 into 20, about 1,500.
No, more.
More.
3,000 plus.
Yeah, we have to say, I did not actually check that number in that fashion, but we can check that. This last year is this one. The 2005 to 2010, we had 130 papers per year average. Now, last to the 2010 to 2014, we have 174 papers per year. So we are about 33% increase in a publication in the last 3 years.
Maybe what's adding here that these papers are not only in number, they also
They are
lot
in
of
high journalism. Nature, Science, Cell. Impact factor is more. And then several Nobel laureates visited Jim Watson, Francis Crick, everybody, George Porter, Paul Berg, Paul Nurse also one of them, Sidney Brenner. All these Nobel Prize winners have visited us in the last 4 years.
So they all have come here?
Yeah, they all come here. All of these people who...
अभी कोई आया है जो इंडियन साइंस कांग्रेस में इधर आया कोई?
No, अभी नहीं है. This time we do not. Yeah. And
but you invite them or they come on their own?
No, we invite them, but also they accept invitation depending on whether you know the institution is well known, so they would like to come. Actually, Francis Crick spent 3-4 weeks with us. Couple of other people also spent a lot of time with us. And this last 3 years, we had these 4 Nobel laureates visit us and give lectures at CCMB. And we also have PhD program, summer training program, other activities. And we have one Hindi magazine called Jignasa. Once in a year we publish for science population in Hindi language. And then we have, when we open day, CCMB we open...
What is the name of the magazine?
Jignasa, that's Jignasa.
Jigyasa.
Jigyasa, yeah. Jigyasa is the magazine, that's once in a year we publish. And this all scientists in CCMB write articles, popular articles in Hindi. And once in a year, CCMB building is open for people to visit. That's open day time. See that crowd of the people coming to see CCMB.
So the average layman, what does he understand in the labs?
We will put up outside posters explaining what it is.
We
in
make
basic
special
language.
poster
Very informal, very understandable
They
to
come
common
inside the labs and everywhere?
They'll first see the posters. We also take them inside the laboratories, and in the groups we'll take them. So we get about on an average about 10 to 12,000 people per year, per one day. But that day, one unique day, on that day we had 21,000 people visit us. So it's very rarely that happens though. And CCMB contribution to CSR is fairly high. For example, entire CSR between this period 2008 to 12 published 5 papers in Nature. Out of 5, 4 papers came from CCMB. As you know, CSR has 38 laboratories. So out of 38 laboratories, only 5 came. Other 4 came from CCMB. In science, for example, 3 were there. In science, the total 5 were there, 3 came from CCMB. So that large number of important papers came from CCMB. And these are certain projects that we are working on. These are 12th 5-year plan projects. And as I just mentioned, those antibiotic vaccine, this Shanvac came from CCMB. We will, I think I'll skip this we can probably go and visit the place. These are certain new findings that we had. So we also looking at the bacteria in Antarctica, Himalayas, other places. Our CCMB scientists have found large number of bacteria in different places. And DNA fingerprinting technology was developed. A new institution is set up from here. There's diabetes. Chandak, you want to tell what it is?
He told just now the same thing, no?
Yeah. Yes, we showed that the phenotype of Indian, which is with less muscle and more of obesity, that we showed that it is programmed very early in life. Right at the conception itself, it seems that happens. And then we found that it is because of the high homocysteine level in the mothers, which is caused because of B12 deficiency. So globally, actually, folate deficiency causes high homocysteine level, but in Indian, it's the B12 deficiency which causes. So this seems that this intervention, if we are able to show its molecular biological function, then probably this intervention can be used as one of the ways that the early programming for diabetes can be put. So as I told, we are currently undertaking an intervention study also.
We have another laboratory called LACONS, Laboratory for Conservation of Endangered Species. There we are trying to see how molecular biology tools can be utilized to preserve wildlife, conserve wildlife. So in this laboratory we also have cages for animals. We have few leopards there, 5 leopards are also there in the lab. So we're looking at the reproductive cycle and see how we can help them survive. And we did some work for Indian Air Force also. So Air Force pilots, when they are taking off flights, the birds hit the aircrafts, and there's a problem for them. So they asked us whether we can help. So we did the barcoding of all the birds nearby, identify what are the birds and what plants, what trees that they use. So by re-engineering the plants around, then now they can protect. And now they have a lot of reduction in that. They said reduction in quantum amount damage to the aircrafts after implementing the modules that we suggested. So that was application of biological knowledge to aerospace area.
Flying up here.
This is the some of the rise that he was mentioning you. That's how the bacterial infection would have come. So it's now being heavily utilized for that. And we are also trying to grow apples in Andhra Pradesh. So apples belong to only Kashmir area, but we are now planning to grow apples in Andhra, in Vishaka area. There is one place in this area where temperature drops to very low temperature, about 2 degrees. So we are trying to grow apples there. And this is a diagnostic tool that I said it's now in the market. And that's a new nanoparticle based approaches that we are developing. That's a fungal keratitis, and you can cure the fungal keratitis, but we need to put drops every 2 hours. So we are trying to see whether we can generate nanoparticle which will bind to the surface and treat it. So we have now made these nanoparticles. That's how it functions. We made the particles. We are now trying on the animal models. This is a photodynamic molecule that we made. That's the structure of the molecule. We can use it for treating cancer. For example, that's a tumor. The tumor that is developing. After tumor develops, leave it, don't do anything, then the tumor within 15 days that will grow that much. But in the second case, we start the tumor and inject the photodynamic molecule and shine light. Then you have no tumor. Completely tumor regresses. So this is working very effectively in this model system as of now. That we have to see how it goes into the real clinical trials. This is our strategy is that threefold work. Curiosity-driven basic science. One is a very important, this is major activity, published in good journals. Second is application-driven high-quality basic science, the one which most of it now we discussed about. And third is the use of existing knowledge to produce products or service of value, innovate. Here we already have existing knowledge. When we have problems, we have knowledge. Link the knowledge to the problems and see how we can provide solutions. So that is new to market, but not necessarily unknown. That's our current strategy to work on 3 dimensions. And we have taken new initiative, as I was mentioning to you. They have conducted a children's program and distinguished lecture series, etc. We started, and in coming few years, of course, we'll continue high quality basic research, and we will be focusing more on stem cell biology, cell-based therapies, plant breeding, agriculture, clonal production, seed production that I did not describe, and molecular marker-assisted breeding. That's one which we talked about. Epigenetics of aging, health, disease, nanobiology. These are the areas that we will be working in for. So our vision is to bring CCMB and Indian biological research to the global leadership position.
अभी पहुंचा नहीं है?
अभी पहुंचा मतलब we are leaders, yes, but not world doesn't recognize us yet as a global leaders. and also to evolve continuously to be relevant to the response to the societal needs. That's one of the things that we do. And we should stimulate to ambience that promotes creativity and innovation. And in addition, ignite young minds so that we will have replacement soon. So these are general ideas. I skipped many of the data points because you will know more when we visit more often to CCI.
इस सारा अगर एक सवाल मैं पूछूं कि जितनी जैसे हाई एंडेड रिसर्च वगैरह हो रही है, तो आप में से कोई भी उसका जवाब दे सकते हैं, उसका प्रैक्टिकल एप्लीकेशन के एंगल से समाज में किसको कहाँ क्या फ़ायदा हो रहा है, उसका कोई एक आध कोई ग्लेरिंग जो सबसे प्रोमिनेंट एग्जांपल हो वो मुझे
वो तो एक एग्जांपल तो डीएनए फिंगरप्रिंटिंग
वो तो है,
एक तो डीएनए
दैट इज नोन टू द पूरा वर्ल्ड को पता
तो
है।
दूसरा ये है कि वो राइस वो जो उन्होंने बना रहे राइस अभी 80,000 एकर्स में वो राइस बोल रहा है अभी, दैट्स राइस इज वन न्यू थिंग एंड द IDC chip that is made now it's in entire India, it is being sold, and also Southeast Asia it is being sold. So now it, as that DNA based DNA based chip for eye diseases, that's now Southeast Asia and India it is being sold. It went into the public.
उसमें आई डिजीजेस के लिए स्पेसिफिक्स में वो जो ग्लाकोमा की बात कर रहे थे वो या कोई
No infections. These infections, eye infections. This only for eye infections.
उससे क्या क्या होगा उससे?
Yeah, you identify what is the infecting organism so that you treat exactly the right way of doing. If you don't treat in
whether it is a bacterial or viral or
not only that, bacterial, viral, fungal is one level of doing. We'll actually go to species level.
Which kind of? Yeah.
We tell the what bacteria, what species. And we extended actually this technology now, it's now going to come soon, is that we are now looking at antibacterial resistance. So tell which organism it is sensitive to. That also we'll do by again same fingerprinting approach, same DNA hybridization approach. So these are 2-3 which are right away in public, and Chandak's approach, Chandak's work will be very critical because if you can tell government that okay add B12 so that we can future diabetic load can be decreased, that will be a massive contribution to the.
But that has not been applied right now.
न्यूरल ट्यूब डिफेक्ट जो है वो फोलेट के सप्लीमेंटेशन से विदेशों में प्रिवेंट किया जाता है, लेकिन इंडिया में फोलेट की डेफिशिएंसी ही नहीं है। तो हमने रिसर्च से ये पता लगाया कि फोलेट के बजाय B12 डेफिशिएंसी की वजह से न्यूरल ट्यूब डिफेक्ट यहाँ होता है। तो हम अभी ICMR वगैरह को भी हमने इंफॉर्मेशन दिया है, हमारी रिसर्च पब्लिश हुई है। तो आप फोलेट तो ठीक है एनीमिया वगैरह प्रिवेंट करने के लिए देते हैं, पर उसमें B12 भी डालना ज़रूरी है, तो उससे एनीमिया में भी प्रिवेंशन होगा और न्यूरल ट्यूब डिफेक्ट के प्रिवेंशन में भी ये हेल्प करेगा। और उसके साथ साथ जैसा मैंने बताया फीटल प्रोग्रामिंग के ऊपर भी इफेक्ट है, तो एक ही इंटरवेंशन से हम 2-3 इफेक्ट को पा सकते हैं। और डॉक्टर घनश्याम ने भी बताया, ग्लाकोमा के बारे में बताया उन्होंने, तो जो जीन जो मोस्टली इन्वॉल्व है, अगर उसकी मॉलिक्यूलर बायोलॉजी अच्छे से पता चलेगी तो वो एक टारगेट या किसी तरह के रूप में यूज हो सकता है।
This is the most important part, is that we know what we want to achieve, so do the basic research towards that goal. Now for example glaucoma is mentioned, what causes glaucoma is still not very clear. If we get understand the exact molecule involved in it, then one can develop the drug. That is, as I mentioned, the process. But as we asked the question, what is already achieved, one thing I was mentioning to you when you were sitting in the room, that is the first recombinant vaccine in India came out of CCMB, which made a massive difference. Hepatitis virus. So that the difference it made is tremendous. As I mentioned, 3,000 rupees wala become 100 rupees, and if you 0.1% of Indian population has taken it, so that multiplied to 2,000, it will be huge number of thousands of crores. So this will be indirect benefit, not really the cash flow, but it cause the benefit.
ये DNA टेस्टिंग जो आपने ये सारा ये फिंगर प्रिंटिंग वगैरह ये सब आपके यहीं डेवलप हुआ है?
हाँ पूरा, everything is developed in कंट्री में। Yeah, कंट्री में नहीं, this lab
नहीं, पूरे कंट्री के अंदर सबसे पहले यही।
हाँ पहले, yes,
1991
first
एशिया में सबसे पहले।
Yeah, this is the third country in the world. This is India is third country in the world. CCMB is the one that place it was done. This is the first time.
फर्स्ट कौन है?
First US and UK. First UK and US.
First UK, then US and then
UK is Alec Jeffrey in UK and then US.
And CCMB had a major contribution in putting it in Indian Penal Code.
Indian Penal Code. Yeah.
Judges
Well,
never
the
used to
most important-
accept
most
that
important
before.
was to be-
Our scientists used to go and talk to them. In early 90s, Dr. Laji Singh did that.
So legally,
मतलब हमारा पेनल कोड में थे ही नहीं,
Yeah. Nobody
डीएनए
knows.
मानते नहीं थे।
yeah.
अभी तो मान रहा है, अभी तो कानून है।
There are a few of them which we can talk about. But many are now in the pipeline which will show up.
what do we do next?
Now we can just walk around and see few facilities.
You want to say?
Thank you so much.
Yeah?
Thank you.
Huh?
आप कुछ
Yeah,
बोलना चाहते हैं?
आपको
नहीं,
बोलना
नहीं,
चाहते
मैंने
हैं?
क्या बोलना है, सब आप
This
लोग
is-
सब साइंटिस्ट लोग हैं।
नहीं, आप
नहीं,
कुछ-
मेरा फोकस यही है कि मेरा ख्याल से सब बहुत अच्छे काम हो रहे हैं। अब थोड़ा सा री-स्ट्रैटेजाइज करना चाहिए इन टर्म्स ऑफ कन्वर्टिंग ऑल दिस इनटू कनेक्टिंग इट विद द पीपल। जैसे कोई चीज़ लैब के लिए बहुत अच्छी है, पब्लिकेशन के लिए बहुत अच्छी है, बट अल्टीमेटली उसका एप्लीकेशन कितना हुआ है साइंस का लोगों के बेनिफिट के लिए, मैनकाइंड के लिए, अल्टीमेटली वो ज़्यादा इम्पोर्टेंट है। मतलब इम्पोर्टेंट तो ये सब कुछ है, लेकिन ये तो जो है ये एक जनरल के रूप में किसी किताब में छप जाएगा और छप के किसी लाइब्रेरी में रखा जाएगा और कहीं किसी जगह शायद वेबसाइट पे आ जाएगा, बट लाभ जनता को उसका कैसे मिलेगा देश की या दुनिया की प्रॉब्लम्स के सॉल्व होने में, इस पे फोकस होके आउट ऑफ द बॉक्स सोच के कुछ नया सोचना चाहिए। जैसे आप सारे लोग इस पे थोड़ा ब्रेनस्टॉर्मिंग, आई कैन नॉट टेल यू कि आप ये करिए तो आपको ये होगा, बट यू नो व्हाट आर द थिंग्स दैट यू कैन डू। उसमें से क्या चीज़ को पहले करना, फर्स्ट थिंग्स फर्स्ट का जो प्रिंसिपल है वो कैसे आप अप्लाई कर सकते हैं। अभी वी आर एट द मतलब एक क्रॉस रोड पे खड़े हैं यहाँ। मुझे लगता है कि बहुत सारी प्रॉब्लम्स को पहले तो कौन सी प्रॉब्लम है जिसको डायग्नोज करके हम उसको सॉल्यूशन निकाल सकते हैं, तो फिर उसकी सॉल्यूशन निकालने के लिए उसके फोकस्ड वे में उसके ऊपर काम करना है। नॉट दैट यू आर नॉट डूइंग इट, बट ये थोड़ा सा मुझे लगता है अप्रोच में चेंज होना चाहिए, क्योंकि हज़ारों पेपर्स भी हैं, हज़ारों लैब्स भी। अभी मैं मिनिस्टर साइंस टेक्नोलॉजी बनने के बाद आई सेट की सबसे बड़ी लैब कौन सी है, तो उन्होंने कहा जोहरट की लैब सबसे पुरानी है, सबसे बड़ी है, CSIR की। तो जोहराट या जो भी है, जोहराट सॉरी जोहरट नहीं,
नीस्ट
जोहराट। तो वो 61 की बनी हुई लैब है। आई वेंट देयर, आई स्पेंड 7 आवर्स। फिर एक गुवाहाटी की एक उसमें डिपार्टमेंट में। बट अगर मैं ये कहूँ कि भाई 39 साल में या 50 साल में 61 से कितने साल उनके 39 से ज़्यादा,
50 से ज़्यादा हुआ,
ओवर 50 ईयर्स हो गए ना। तो जैसे मैंने आपसे ये सवाल पूछा, ऐसे सब जगह हम पूछने की पता करने की कोशिश करते हैं भाई इसमें से कोई ब्रेक थ्रू चीज़ बताओ, ऐसी चीज़ हमको बता दो जिसके कारण यू आर ऑल्सो प्राउड ऑफ दैट एंड द कंट्री कैन बी एंड द वर्ल्ड कैन बी, और उससे लोगों का भला हो। जैसे आपने DNA फिंगरप्रिंटिंग बताया, इट इज़ समथिंग फॉर विच दिस इज़ ए प्लेस विच इज़ रिकॉग्नाइज्ड, उससे बहुत सारे लोगों का भला वाला भी हो रहा है और सारा हो रहा है। मैं खुद आई ऑलवेज फील ग्रेटफुल टू दिस लैब कि मैं 96 के अंदर जो है मेरे को वो पॉलिटिकल कॉन्स्पिरेसी के अंदर मुझे जो है डाल दिया। मैंने बोला अब साहब मेरा वेसेक्टमी हुआ हुआ है, तो उसके बावजूद भी मेरे पीछे लिखते थे, रोज़ आर्टिकल लिखते थे। एक आदमी ने आर्टिकल लिख दिया कि चिल्ड्रन कैन बी प्रोड्यूस्ड इवन आफ्टर वेसेक्टमी। देन one of my friend came to my rescue. He wrote a letter to me. He was my friend in medical college. He said, "Buddy, इनको you should give a befitting reply to them. There is a technology now. We can do DNA testing and all that." So, but the police never said. Police said कि साहब हम we have no case against you. We don't want to do your DNA test and all. So I had to take the help of the senior leaders to convince the police to get myself subjected for a DNA test. Rather, lot of leaders they got angry कि यार आप ये प्रॉब्लम खड़ा कर रहे हो, DNA टेस्ट अपना करा के जो है आगे हर एक आदमी हर एक पॉलिटिशियन का DNA टेस्ट डिमांड करेगा। तो आई रिमेम्बर, अभी तो मैं आज पहली बार देख रहा हूँ ये लैब है, तो उस ज़माने में 96 में आई थिंक
दिस वाज स्टार्टेड 89
नहीं 89 नहीं,
हमने यहाँ पे
व्हाट आई एम टॉकिंग अबाउट इज 96। तो आई वाज ए मिनिस्टर। तो इधर डीएनए टेस्ट, अब डीएनए टेस्ट का रिपोर्ट होएगा उसके बाद भी कैसे कोई क्या बोलेगा, तो फिर नहीं ये नहीं होगा वो होगा, मैंने कहा उसका भी कर लो, वो होगा उसका भी कर लो, जिसका जिसका करना है कर लो. तो दैट वे दिस इज़ अ वेरी गुड थिंग इन द सेंस दैट यू कैन हेल्प.
It has stood the test of time also, yeah.
तो ये चीज़ जो है, तो उस तरीके से आई फील कि फोकस हमारा मतलब थोड़ा रिवाइज करने या रिव्यू करना या अभी ब्रेनस्टॉर्म करना या आउट ऑफ द बॉक्स जाना. रूटीन से जो कुछ हो सकता था वो तो हो गया है. अब 67 ईयर्स के बाद इट इज़ टाइम टू कम आउट ऑफ द बॉक्स. उसके बारे में आपको रेगुलरली थोड़ा ब्रेनस्टॉर्म करना चाहिए, मुझे ऐसा लगता है. मोस्ट ऑफ द आइडियाज़ दे कम आउट ऑफ ब्रेनस्टॉर्मिंग. हम लोगों ने पोलियो एरेडिकेशन किया तो वी स्टार्टेड विद ए स्मॉल ग्रुप ऑफ 4 people ओनली, बट वी यूज्ड टू ब्रेनस्टॉर्म अबाउट एवरी प्रॉब्लम. बहुत बड़ी प्रॉब्लम आ जाती थी तो किसी मैनेजमेंट इंस्टीट्यूट के डायरेक्टर को फ़ोन करते थे, वो 2 घंटा अपने बच्चों से ब्रेनस्टॉर्मिंग करा के हमें सॉल्यूशन दे देती थी. दैट्स हाउ वी स्टार्टेड व्हेन वी बिगेन. उसी में से बहुत नए नए आइडियाज़ आते हैं, छोटे छोटे इनपुट से मैक्सिमम आउटपुट बाहर निकल जाते हैं. तो दैट इज़ व्हाट आई फील कि बाकी तो आप लोग साइंटिस्ट लोग हैं, सब जानते ही हैं, मैं क्या बताऊंगा आपको. चलिए लेट्स गो. थैंक यू. चलिए आइए.
So do you want some more that will become I'll just start it. These are the That's the mission which will hopefully being made. These are the older machines, they're important. This is the wound healing assist. How the wound is cells are dividing and moving, healing the wound. Cells are coming together, closing the wound. Now we'll measure how much time we put to close the wound. It is about 24 hours this one. So we'll measure how long it takes. And then layer over. If we know cells are present differently, then we need a scratch. For the scratch, we create a wound. After wound is moved, then we put it on the types of microscope. And the cells will divide slowly. This is a time-lapse microscope system. So it took 24 hours. You can see the cells are dividing. And this is maybe some of the that's some of the hard this is something else they are showing. This is the normal cancer process. When they come together, they should stop. If they don't stop, they grow. Without stopping, then it becomes cancer. Now we are trying to understand when the cells contact, how they stop dividing, and how the motility is also moving, how the motility comes. Because primary cancer is one place, secondary cancer is a different place. The cell has to move there. If you can arrest the movement, then you can prevent the secondary cancer. So we are looking at how they fall out, what are the important molecules. This atomic force field microscopy. We can also do that. This is very, very high resolution microscope. We can say nanometer structures. Nanometer structure. Medicine seeing the structure, what's the chemical composition of that? Also we can find out by drawing. You shine light on it. What are light is coming out? From that you can analyze. You can identify what type of material. Is it a protein, DNA, carbohydrate, what it is. That also can be. You can do spectroscopy and imaging together. Very subsequent, this is useful for nanobiology, nanotechnology application in the field. So when we put the sample, we close it for vibration free. These have to be removed from different scales. Two diagrams. This is the multi-photon. Engineer. Good. Engineering also science, medicine also science. All can be done. To become physical scientist. Okay. Initial scientist also. These people are getting into the lab now. They are young scientists. You see their aprons, they are wearing the aprons, everything, and they are doing experiments here. They are very young scientists. Okay.
Wish you all the best.
Join, we should take a
All of you grown-up children, come to one side.
Actually, transfer to the sample, and we can see the internal organs so we can further magnify it.
This is 200 electron volt.
Actually, we can the 2.4 angstrom we can resolve by the system.
Very high resolution matrix.
How old is it?
Good.
Yeah,
it's good. Now, old one is 25 years old one. We kept in the open area for children to play with.
Because children are very interested in seeing an electron microscope. The first thing they ask is where's an electron
So this is like an MRI machine where we get a very high resolution. So we look at the baby growth, and identify the diseases at a much earlier stage before they can be diagnosed using clinical markers that are available. So for example, there is a rat. We can put the radiation right here, and after that we put this probe inside, and when it comes, then they actually this NMI spectrum is nothing but we take the spectrum from proton. So what is they mainly proton. So different protons in our proton depending on the NMI case, the distribution of protons will be different. According to that, we will get different kind of things. Here you can see the case of this MPTPJ toxic for this brain. With MPTP determines a large number of kind of defects, and lower the lower is vascular disease. So we can image different kind of disease. One of the applications we are just coming about Alzheimer's disease. There is no diagnosis for Alzheimer's disease. Now what is because is Alzheimer's disease, glucose metabolism rate is changing even before the disease comes up. So now by studying glucose metabolism rate in the NMR spectroscopy, one can predict that after 5-6 years you might get the disease. Treatment time will start with this group, but treatment is like a 9 by maybe a little stress proteins. This but stress, we actually another question is if you put this stress proteins alpha beta B2 they dissociate the aggregations because the aggregations are non-coding RNA. So we are trying to understand the structural and mechanistic difference of the processes in which non-coding RNAs are involved. For example, my lab has solved the structures of proteins in solution. So advantage of this technique is finding the structure of a protein physiological condition. So like how it would be in the cell and how it would function in the cell. Also another advantage that we have with this technique is that we can understand the dynamics of a molecule. So once we have a drug, for example, let's take somebody takes a drug, it does not go and bind some receptor changes its composition. And those changes in the composition we can find out. For example, this is one of the largest structures solved in this country using this machine. 32 protein structure we have solved. 32 kilodalton size of the protein. This is a protein called a CSIR-Cellular. This is the largest protein that we solved from this country using this kind of a technique so far. And we can also look at some of the domain motions. So this is an alpha helix of a protein. This is the alpha helix structure of a protein. And there is a beta strand that you can see over here. This is the organization of amino acids. The organization of amino acid folds into two secondary structure elements, what we call it as
आप यहाँ देख सकते हैं ये अल्फा हेलिक्स होता है एंड द मोमेंट RNA comes and binds, RNA binds to this patch, these three residues over here from these structures.
This was actually my original field of activity was protein structure. Then I became more on medical side. This is, you can see looking from this outside. Those are our other buildings. These are our source. This is the workshop. There's a source in this area. And this is our this is our canteen. And these are the house. So we have nice.
We have developed a technique where 5 to 6,000 proteins can come at a one. That's help us because whole human body is 20,000 proteins. So idea is to characterize each and every protein of the body. Then major focus would be that for disease. So like why, how, biomarker. And then we develop a drug. But how drugs target, that's the other thing we have to use for clinical.
This is the DNA. This is the DNA.
DNA sequencing lab.
For DNA sequencing work is done.
But right now we are also just to take the samples and do the analysis. The CCMB samples are also done.
What we do is these machines can sequence DNA. But DNA first experiments are done in the laboratory. Then the piece has to be given to sequencing. The sequencing part, we have machines here, we are sequencing organs. But now work is so much. What we thought is that in Saturday, Sunday machine, this time is free available. So now outsourcing company. Those employees are working here. They will sequence for our samples as well as outside sources. So they will get money from outside sources. So that much will be getting very good. And we also don't have to employ the staff to the salary or the maintenance
Because a lot
also will
of
take it.
samples can